The catalase is the enzyme that catalyzes the breakdown of hydrogen peroxide to form water and oxygen. And here they have given us different, there are 3 cubes of potato tissue. What we have to do, we have to investigate how the surface area of potato tissue affect the activity of catalase. And activity of catalase is measured by the height of form, which is written over here. Okay, now tell me what is dependent variable and what is independent variable. Surface area. Read again. And how do we measure the activity? Red, height of the form. Okay. So the dependent variable is height of the form and independent variable is the surface area. Okay. There. But when we have to give the readings, so that we cannot like judge it by looking at the enzyme. We have to take some like measurable unit, and that is the height. Okay, now here is one cube, and keep in mind cube is a 3D shape. It has 6 faces. Okay, so surface area is based on the 6 faces. Let's say it's L square into 6. Okay, so they have given you the surface area, which is 6 centimeter square. Okay, in the other part they cut them into half, and the surface area turns out to be 8 centimeter square. Okay, again they cut it in more pieces. Now you have 8 pieces. 1 2 3 4 5, like 5 6 7 8. Okay, they have told us that they made 8 pieces, and you have to calculate the surface area. So the surface area of one cube, that is equal to 6 into L square. We have done this. Okay, and multiply by 6 is 1.5. Okay, this is for 1 cube, but now we have to do it for 8 cubes. Then we have to multiply this with 8. So you have to do all the working over here. 8 into 1.5 centimeter square, that is equal to 12. Okay, yes, this is for 1 cube. Okay, we found the surface area of 1 cube, and then we multiplied with 8 cubes. Okay, let's go back to the practical. Okay, this is the surface area, how we change the surface area. Now they use 3 test tubes labeled as 1, 2 and 8, and you have to use the same labeling that they've used in question. Then they use 10 cubic centimeter of hydrogen peroxide into each of the test tube. It means this one is your control variable, which is constant. Put 1 of the cube of potato in test tube 1. Put 1 of the remaining cube of potato onto the white tile. Use the ruler and scalpel to measure and cut the cube. This will make 2 equal sizes, and the total surface area is this. Okay, now they have used ruler and scalpel as well. Okay, next one is they have used the other cube, and they cut it into more smaller pieces, and you have to calculate the surface area. After cutting them, they start the stopwatch and check the activity in test tube 1, 2 and 8, and the given time is 5 minutes. Okay, after 5 minutes, the distance or the height of the form is calculated and the reading is recorded. Let's say what's the reading. So you have to take the reading, but keep in mind when you take the reading of form, this should be from the top bubble. Use the top bubble to find the height. Okay, whatever your height is, you have to give the readings over here. Sorry. So there would be two col. Oh my god. Either I'm not sitting right. This way. Okay. So we'll make two columns. Sorry, it's not working for me. Okay. For first one, we'll label it as test tube 1, 2, and 8. And for second one, height of the foam and what unit you are going to use. Okay, you can use millimeter, you can use centimeter as well. I would recommend centimeter as they have taken the surface area in centimeter. Exactly. Okay, we use millimeter most of the time, but if in question centimeters are given, then you have to use the centimeter. Okay. Question. Exactly. Okay, state a conclusion of these results. Okay, results are over here. What is your conclusion? More the surface area or greater? Good. Greater the surface area, higher would be the height of foam and more activity of catalase. Okay, which variables were kept constant in this investigation? We are done over here. The concentration of, read it again, hydrogen, the volume of hydrogen peroxide is, okay. What else? Do you have a soft copy of this paper? Okay, let me send you then. Yes, time was 5 minutes. Okay, here is the paper I've sent you. Okay, so the volume of hydrogen peroxide solution, one safety hazard, yes, hydrogen peroxide is toxic. You can write that. So what you need to do? Wear gloves. So hydrogen peroxide is toxic. Wear gloves. Yes. Okay, then the next one, there is another error that they're using scalpel. Okay, so scalpel is a knife. So while using the scalpel, get the piece away from your body. Okay, two possible errors in this investigation. The first error is form. Okay, exactly. So you cannot have the correct reading. Instead, what improvement can be made? We can use a gas syringe for the collection of gas. Okay. Form gas syringe can be used to collect gas or to have a volume of gas. Okay, what can be other error? While cutting the pieces, it may happen that the size may differ. Okay, while cutting, the size may vary, so we have to use the precise, you can say, measurements or scale scale can be used to have the correct measurement or the precise scale. Next one is iodine solution can be used to show the potato's tissue contains starch. What is the positive test with iodine solution? Okay, so iodine solution can be used to show the potato's tissue contains starch. What can be the positive result? Blue black. Okay, the color changes to blue black. Okay, next one is a student investigate the effect of, now this is an other experiment, investigate the effect of copper sulfate on you can use a precise scale to have a cutting while cutting the pieces. Okay, okay, so the result was it turns to blue black. Okay, a student investigated the effect of copper sulfate concentration on the activity of catalase. Concentration on the activity of catalase in potato tissue. So what is the independent variable and what is dependent variable, since they have asked you? Okay, here's the whole experiment. So they have used wait, since I have given you the full statement, they cut the potato disc, and after cutting the potato disc, it was given some time so that it can come to the top. Okay, so that would be your another, this statement. Okay, so what is your dependent variable and what is your independent variable? Correct. Wait, no, I'm writing wrong, sorry. I thought it's independent written. Time taken for the disc of potato to reach the, you have to write the complete statement, okay? Surface of hydrogen peroxide, okay? And the independent variable is the concentration of, concentration of, read it again, copper sulfate solution. Copper sulfate solution. What is the suitable control for this? Wait, you are testing for copper sulfate, so what can be your control? So instead of using a copper sulfate solution, you can use distilled water. Okay. No, you have to tell what should be added in a controlled experiment. Okay, but the volume should be same for same volume. Okay. Oh, copper sulfate solution, I have written surface area. Okay, next one is a graph is given, you have to draw a proper graph. So now here is the main thing. How many readings do you need? You need it till 200, let's say, roughly. And how many boxes do you have? We have 1 box, a 2 box, 3 box and 4 box roughly. Okay, let's divide it at 4, so it would be 50. Okay, so each big box would be of 50. Okay, this is how we determine the scale for x-axis and y-axis. Keep in mind that on x-axis we take the independent variable and on y-axis we take the dependent variable. Okay, so let's say time taken for a disk, time taken for a disk of potato to rice, and unit is second, and the other one is the concentration of copper sulfate solution with unit. So concentration of copper sulfate solution, mole per cubic decimeter. Okay, now how to choose the scale over here? So we have a reading till 0.025. okay, and how many boxes do we have? We have 1 box, 2 box, 3 box, 4 box and 5 box. Okay, so we divide it with 5, it would be 0.005. Okay, so we write the exact 3 units: 005, 010, 015, sorry, 0.020 and 0.025. okay, this is how we determine our scale. I guess this thing is okay for you. Okay, now you have to make a graph, and they have told you have to plot a line graph. Okay, this is not a best fit graph, you can make a simple line. I would recommend make a ruled line instead of making it curved, like join all the points with the help of scale. Okay. Yes, you can. Otherwise they would mention that you have to make a ruled line or a bar graph or anything else. Okay, keep in one more thing I want to tell. There is difference between bar graph and histogram. In histogram the bars are joined, okay, space, and they should be equally spaced. Like I would recommend give space of 5 boxes, then make a bar of 5 boxes. Give space of 5 boxes and make it like it for 5 boxes. Okay. Okay, so this is how your graph looks like. If you want to mention 0.03, you can mention it over here. Okay, since it's in the reading and it's not in 0.003. okay, it would be after 6 boxes. So the reading is 5 over here, so you can like put reading. No, reading is I guess 45 over there. Yes, 45. So it would be here. Okay, done. I'm trying to like add something over here, but I'm not able to. Let's see if I can. No, I cannot. Okay, let's do it later. Wait, what was that? I'm sharing another screen to show you the graph, to have a clear picture of it. So one more thing I want to tell you, since I've asked you to make a line, let me just switch the screen. Okay, here. So I have told you that you have to make, like if unknown value is asked, they would ask you that what is the value over there or what is the value over there. So you have to make straight lines from the x-axis and y-axis and the point where they are coinciding. Okay, these lines should be very clear, since they would ask you that what is the reading over there or what is the reading over, like any point they can ask. Okay, it should be You have to write 0.5 then. You have to. Okay, let's go back to this. Okay, now the next question is you have to make, oh sorry, whatever is your estimation you have to give over here, that is for 80 seconds. So 80 seconds lie over here up till, sorry, it would be like 6 boxes up to over here 80. Okay, okay, next one is you have to make the diagram. In making diagram, it should be very simple. It's a 4 marks, so 1 mark is for stock and 1 mark is for over outline. The size should be bigger. There should be this midline and number of seeds, 5 up 5 down. Okay, if a seed is not straight, it's in some other direction, you have to give it a place. Okay, you have to make a proper diagram. Done. Okay, if the diagram is for, let's say, 4 centimeter, then you can draw it for like 7 centimeter. Okay, make it bigger, make it clear. So there are 5 seeds up and 5 seeds down. Keep this thing in mind while making a diagram. Okay, next one is you have to calculate the length of PQ, and the length of PQ turns to be, I guess it's 7.4. okay, on our scale. So when how do we convert 7.4 centimeter into millimeter? 10, we multiply with 10, so it would be 74 millimeter. Okay, so how would you calculate the actual length? Actual length is equal to the wait actual is equal to the image size or the length of PQ over magnification. So it would be 74 divided by the magnification given is 1.7. So to divide it with 1.7, so okay, so how many figures are required? 3. So okay, so your answer is correct, 43.5. okay. Okay, the next one is Benedict solution. A Benedict solution is given. What could be the positive result for Benedict solution? Good. Okay. Yes, blue to brick red. Blue lilac. Yes, for Burette reagent is blue to lilac. Okay, the next one is you have to calculate the percentage increase or decrease. Since such questions are not there in paper, like for you it's not in paper 2, and for IGCSE it's not there in paper 4, so there are chances that such question could come, and there are more chances that there can be questions related to dichotomous key or the reproduction in plants, since they are not like in P4. So they may give you such questions over here. Okay, so let's calculate the percentage increase. So the initial sugar content is 19.6 and the final sugar content is 36.1. So it would be equal to final minus initial divided by initial multiplied by 100. So it would be 36.1 minus 19.6 divided by 19.6 multiplied by 100. So you have to give the answer in 2 decimal places. $84.18, correct. Okay. How to plan the investigation to determine the effect of speed on the rate of transpiration? I guess you have done this question already. No, it was in the O-levels exam. It was there. It's from O-levels. There is already such investigation in your papers. Okay, let's do it. Let's do it. Okay, how to determine the effect of wind speed on the rate of transpiration? Let's find out what is independent variable and what is dependent variable. So the and how would you determine the rate of transpiration? Potometer, yes. Okay, and the potometer would be used, but potometer would be used. How would you determine the rate of transpiration? Okay, we determine the activity of catalyst with the help of height of foam, right? So, wait, the distance traveled by the air bubble in the potometer. Okay, you need to revise potometer topic, okay? Please revise it. Okay. Okay, so what you would do, you would determine you would select 3 different wind speed. You can use fan and put it at different distances, or you can change the intensity of fan. Okay. Okay, the next step is how to set a potometer. You have to take a leafy shoot. Okay, let's say 2 to 3 different light intensities, wind speed would be used by placing a fan at a different distance from the potometer. Okay, this is how you would determine your independent variable or set up your independent potometer. Okay, now for rate of transpiration, you would take 3 same leafy shoots. Since it's transpiration and it's related to stomata, so try to have equal number of leaves on each shoot. Okay, leafy shoot, not same, let's say similar. The word is similar. Leafy shoots would be taken and set up in a potometer, and if the potometer, there is no need to give the whole thing. How to set up the potometer, that's a different whole new experiment. Okay, just say that you have used the potometer. Okay, note down or record the initial reading for the for the air bubble would be recorded. In a table, you can write in a table, or otherwise would be recorded is enough. Okay, what should be kept constant? Now, transpiration is affected by 4 factors. One is given wind speed. What are the other 3 factors? One is temperature, light, and the last one is humidity. So these 3 factors should be kept constant. Okay, so let's say yes, temperature, light intensity, humidity, number of leaves on a shoot, what else can we write? Should be no, since we have used the should be kept constant. Okay, now after a given time, or let's say what was I was trying to write? Okay, the wind speed, the fan was turned on let's say the fan would be turned on, and after a given time the distance traveled by the air bubble would be recorded. Okay, traveled by the air bubble would be recorded. Okay, now after this what would you do? Repeat, first of all repeat the experiment at least thrice. Okay, how to conclude it? You would take, yeah, mean value. Wind speed, higher the wind speed, higher would be rate of transpiration. Okay, what, okay, what if they ask you about the precaution, since you are cutting a shoot, you are cutting shoot, okay, so scalpel, okay, moreover you're using fan, so it's an electric fan, so you have to be careful while using a fan. Okay, so no, but we have to give, like, we have to use those terms while we are giving a method. You have to tell in method what apparatus you're using. We are using potometer, that we have mentioned. We are using fan, that we have mentioned. We are using the chute, we have mentioned that. Okay, yes, we have mentioned all the apparatus, but we don't give a simple heading that this apparatus is going to be used. Okay, so this is all for this paper. Let's move to the next one.